All right, BIM 100 students, welcome. In this explainer, we're going to crack the code on Lecture 11, Eukaryotic Translation, and get you totally dialed in for Professor Pasquinelli's final exam.
Look, I know translation can be one of those really tricky topics, but let's think of this session as your secret weapon. We're going to go through it step by step to build your confidence so you can walk into that exam and absolutely nail any translation question that comes your way.
So here's our game plan. We'll kick things off by meeting the cast of characters. Then we'll decode the genetic scroll to make sure we understand the language. From there, we'll jump into the translation incantation itself, check out what happens when spells go wrong, and we'll finish up with some key Pasquinelli's puzzlers to make sure you're ready for the test.
Okay, let's get right into it. Before our star player, the mRNA, can be translated, it's got to assemble its crew. So let's meet these key players.
It all starts with the mature messenger RNA. You really want to think of this as the master blueprint. It's fresh out of the nucleus, all processed and ready to go, and it contains the precise instructions for building one specific protein.
Next up, we have the transfer RNAs, or tRNAs. These little guys are the brilliant translators of the cell. They read a specific three-letter codon on the mRNA and bring in the one and only one correct amino acid that matches it. They are absolutely essential for making sure the right protein gets built.
And this is where all the magic happens, the ribosome. The ribosome is basically the factory floor for protein synthesis. It has these three crucial docking stations, the A, P, and E sites, that perfectly orchestrate the entire assembly line.
All right, so we've met the team. We've got the blueprint, the translator, and the factory. Now let's take a closer look at the actual language written on that blueprint, the genetic code itself.
The code is actually pretty simple. It's always read in three-letter words called codons. Now there are two key properties you got to remember. First, it's unambiguous, which means one codon always codes for one specific amino acid, no exceptions. But it's also degenerate, which is just a fancy way of saying you can have multiple different codons that all code for the same amino acid. And of course, you've got your starting signal, AUG, and three stop signs.
Okay, let's talk about the wobble hypothesis. And you are definitely going to want to star this in your notes. This is a huge concept for the exam. Basically, it means the base pairing rules get a little bit flexible, but only at one specific spot. It's the third base of the mRNA's codon that can form a wobbly pair with the first base of the tRNA's anticodon. Here's your pro tip. A classic mistake on the final is getting this backwards, so remember, it's the first base of the tRNA's anticodon that wobbles.
And here we go. This is a perfect example of a classic Pasquinelli-style exam question. You'll get a long mRNA sequence, and she'll ask something like, what amino acid is encoded by the 12th codon? The trick here isn't to get intimidated. You just have to be methodical. Carefully count your codons in groups of 3, find the right one, and then just look it up on the genetic code chart.
Okay, so we've got the players and we know the rules of the language. Now we're ready for the main event, the translation process itself. And it all happens in 3 key steps: initiation, elongation, and termination.
First up is initiation. This is basically the setup phase. All the machinery, the small part of the ribosome, and that special initiator tRNA come together at the 5' end of the mRNA. Then this whole complex just slides down the mRNA, scanning, until it finds the starting line, that first AUG codon.
Next is elongation, and this is where the protein actually starts getting built. It's like this constant, repetitive 3-step dance. A new tRNA carrying its amino acid comes into the A site, a peptide bond is formed, and then the whole ribosome shifts one codon down the line. And the cycle just repeats over and over and over, adding one amino acid at a time.
And finally, we hit termination. This whole thing is triggered when the ribosome runs into a stop codon in the A site. Now remember, there's no tRNA that recognizes stop. So instead, this protein called a release factor comes in, it cuts our newly made protein free, and the whole factory just disassembles. The job is done.
And this is why maintaining that reading frame is so critical. Just look at this example. In the original sequence, everything's fine. You get the protein you're supposed to. But on the right, just deleting one single nucleotide. It's a total disaster. It causes what we call a frame shift. The ribosome starts reading the wrong groups of 3, and every single amino acid after that point is completely wrong. You get total gibberish.
But what if the problem isn't a slip-up during translation, but a flaw in the mRNA blueprint itself? Well, the cell actually has an incredible quality control system for that.
It's a system called nonsense-mediated decay, or NMD for short.
and you'll want to know this one. Its whole job is to act like a cellular inspector, finding and destroying any mRNA that has a stop codon where it shouldn't be. This is super important because it stops the cell from making shortened, non-functional, and potentially even harmful proteins.
So how does the cell actually know an mRNA is faulty? Well, a really important mechanism here is the exon junction model. After splicing, a little protein marker called an exon junction complex gets left behind where two exons were joined together. Now, during a test run of translation, a healthy ribosome will just knock all these markers off. But if it hits a premature stop codon, the ribosome falls off too early, leaving a marker behind. That leftover marker is the red flag that tells the cell, hey, this mRNA is bad news, get rid of it.
And this brings us right back to why this stuff matters for your final. This slide shows a real question where NMD was the correct answer. This is proof that this is a super testable concept. You have to be able to look at a mutation and figure out if it would create one of these premature stop codons that would trigger this whole quality control pathway.
All right, let's bring it all home. We're going to do a super quick rapid-fire review of what we're calling Pasquinelli's puzzlers. These are the absolute must-know concepts you need to have locked in for the exam. Okay, for your final study push, focus on these 4 things. Number 1, the wobble rules. Know it's the first base on the anticodon and the third on the codon. Number 2, reading frames. Know how to find the start codon and understand the devastating effect of a frame shift. Number 3, the 3 steps. Be able to walk through the key events of initiation, elongation, and termination. And number 4, quality control. Understand what NMD is and how that exon junction model works. If you've got these 4 down, you are in excellent shape.
And that's it. That's your high-speed tour of eukaryotic translation. You've got the concepts, you know the players, and you understand the process. You are totally ready for this. Now go crush that final. Good luck!